Well, anyway, thank you so much, Habib. And yeah, so I'll tell you a little bit about some work that we've been doing over the last few years that kind of has at its core this idea that we can uh, put together that we can put together different data sets and modalities to get a more complete picture of the brain. Oh, and so I think you know, our day-to-day kind of lives in the lab revolve around this basic problem that we are in the business of generating brain maps, whether we're doing, cortical thinning in a disease, task activation with fMRI, with some cognitive manipulation, whatever. We have this brain map. And what we ideally want to know is, does this brain map look like anything else that we know of, any other biological feature? Does it co-localize with other measurements of structure and function? And when I say that, I mean, does it look like maps of gene transcription of different neurotransmitter receptors, densities of different lamina, things like this, metabolic capacity, and so on. And it's not as though we don't know how to measure these things or put these quantities in the brain. We actually do. There are numerous imaging and tracing technologies that allow us to actually derive most of these features, some with just garden variety MR scans that we do in our, in our centers, and others with technologies that are kind of specialty domains of other institutes around the world, but that make their data freely available. And so what we've been doing over the last few years is coming up with ways to put these things together. So I'll tell you a little bit about some of the tools that we've developed that hopefully are maybe interesting, potentially useful for you guys as well. I'll show you a few examples. We didn't start out making these tools just for the sake of making them, but really we wanted to answer very specific questions. So I'll tell you a little bit about that, and I'll tell you at the end. I'm going to focus the first kind of two parts just on brain maps more generally, but then I'll go into more into connectomics and how we can actually use these maps to build richer representations of brain connectomes that we call annotated connectomes. So just to talk a little bit about the tools, and there's many ways for me to approach this, but the easiest is to just tell you about this latest and greatest thing that we have in the lab that we call neuromaps. It's kind of like a Google Maps for the brain, and it puts together a lot of these features into one place, and you kind of get a sense of what I mean. So the toolbox, it's a Python-based toolbox. The idea is that you, as a user, would come in with a brain map that you have, and it has kind of three components to it. You don't have to use the three components all in sequence or all together. You can use them as you choose, but basically we provide a suite of tools for transforming among the major coordinate systems in brain imaging. So say that you have a map and you want to compare it to another map that's in a different space. How do you put them all in the same space? Well, we'll give you the transformations. We have a curated library of different measures of brain structure and function, all in their native space, so that you can do these comparisons. And finally, we provide a suite of rigorous statistical tests that are basically null models for spatial correlations that take into account spatial autocorrelation in brain maps. I'll talk about that as well in a minute. And then at the end, if you kind of go through the entire sequence, you can get an enrichment score. So you bring in your brain map, and then we tell you, look, this brain map looks a lot like gene expression for these genes, or this type of metabolic process, and so on. But I'll kind of take you through each of these components now, just to give you a sense of how they can be used. So the library is kind of the most interesting one in a way. Basically, we've gone ahead and put together various measures of, it's kind of, we call this microstructure, but you can come up with any other name as well. But this is both from MR and PET, different measures like T1, T2 that everyone knows and likes, but also the PET tracer UCBJ, which is a measure of synapse density, and so on. We have, again, for PET and MRI, different measures of metabolism, so regional differences in metabolic capacity. You can do, and I'm sure you guys have seen these papers where you can take either monkey brains or baby brains and try to warp them into an adult brain. So you can have a measure of how much do different brain areas expand, either over evolution or development. So we have these kind of Evo-Devo expansion maps. We have also both from source resolved MEG and from intracranial EEG, we have maps of power for the canonical electrophysiological frequency bands. We also have the famous intrinsic timescale map from BOLD and various other measures of dynamics as well. We've cross-referenced everything to the Big Brain Atlas. This is the Alan Evans 10 micron Merker stain cell density atlas. So basically what they have there is maps of the thicknesses, kind of cell density in the 6 canonical layers of the neocortex. And we also have, using cell type deconvolution from microarray gene expression, maps of different cell types. The coolest thing, though, that we have, and the one thing, if you'd asked me, what do I want if in the course of my own work, what's the microarchitectural feature that I would most want to have about the brain? It wouldn't be any of these. What I ideally like is a measure of the neurotransmitter receptors. Why? Because neurotransmitter receptors modulate the link between structure and function. Also, they are the primary interface through which we can pharmacologically manipulate brain function. So by the way, at the bottom, I'm occasionally going to show you what some of the lines of code would be to actually run some of this stuff. The only purpose for that is just to show you how simple it is to actually do this. But neurotransmitter receptors, really interesting because you can actually measure them with PET. The only problem is, for some reason, PET researchers don't really talk to each other all that much, and they tend to collect smaller samples because it's obviously very expensive. It's sort of semi-invasive. So what we started to do about 2-3 years ago is we first cold called people that were in our center, and then eventually friends of their friends and other people that were recommended to us across the world. And we basically collated a data set of different PET tracers for different neurotransmitter receptors. That's what this thing actually looks like. So it's a 3D, it's actually whole brain. It includes subcortex as well. And Chemo Architectural Atlas, we have data on well over 1,200 participants and now it's actually not 19 receptors, but actually 20 receptors even, across 9 different neurotransmitter systems. So they are all included in neuromaps as well. And we also have a contribution pipeline. If you have a brain map that you really like, you can send us a note and we can include it. Also, just take a little detour to tell you that I mentioned gene expression a couple of times. We have a tool that came out a year or 2 before that was called Abogen that works seamlessly with neuromaps. So if you're interested in more specific genes and their maps, you can use Abogen. So it'll basically download and pre-process the Allen Atlas microarray data with 1 line of code. It'll parcellate it for you. It'll work, it takes right around a minute to do the whole thing. It can do all sorts of things like allow you to focus on single genes. You can mask out a region of interest. It'll work with any atlas that you give it. And we can also do dense interpolation because the microarray probes are obviously, the gene expression is sampled in a spatially discrete way. So we can get whole brain maps. So basically, if you want a map of a very specific gene, like ApoE4, whatever, MEPT, that sort of thing, you can get it. And then it's kind of a little bit cheeky. If you, it'll also actually write your method section for you. So depending on the pre-processing choices that you pick, it'll spit out this piece of text that'll say to people exactly how you chose to pre-process the data. The idea being that actually we noticed that a lot of people were getting very different results sometimes in the gene expression literature, and it wasn't clear why. And the reason is that people were doing things slightly differently. Different groups were doing things slightly different. So it's kind of an effort to try to get the field to standardize their workflows a little bit. But anyway, so that's the library. And as I said, we are very careful. This is like an artisanal, hand-curated library where we have everything in its native space. We didn't want to screw around with any of the maps, so they're unadulterated. What that means is that if you want to compare maps to one another, you're going to have some way of transforming and going between the coordinate systems because obviously not everyone does things the same way. So, and even though this is kind of like almost, it was sort of an afterthought in a way, in our lab at least, this is probably the most useful and most widely used part of the toolbox. So we provide support for MNI 152, Civet, FS average, FSLR. And if you want to go volume to surface, we've implemented registration fusion. And if you want to go between the different surfaces, we've implemented MSM. We didn't invent registration fusion or MSM, but we did extensive tests to see among the many different ways that exist to do this, how well any of them perform. And so we settled on these two, and they really do work as well as advertised. So basically now you can either take your source map and transform it to the target map space or vice versa, and so on. Or, you can, for whatever else that you're doing in your own work, you can just use this module. We also provide support for parcellation, so you give us any atlas and we'll, it's just one line of code to parcellate a brain map. Okay, and then the last thing is, so you have two maps, you've transformed them into the same space, you want to know if they're similar to one another. What you're going to end up doing is running a spatial correlation between them. This is like probably the most common analysis that we do in the lab. And the problem is that anytime you correlate two brain maps that are spatially autocorrelated, you're going to get inflated p-values. This is something that was fortunately noticed or caught by the field very early on. And I just want to give you a sense of the scale of the problem. So these are two brain maps that have generated that are completely random. They're noise. Their correlation is 0, as one would expect. What I'm going to show you now is what happens when I smooth them out. I apply a smoothing kernel to both of them, and you start to get something that actually looks a lot like the brain maps that we have in brain imaging. They're still completely random, but because they're smooth and I've introduced spatial autocorrelation, now you get very, very high correlations among them. They're obviously not real. And the reason for this is that when you have greater spatial autocorrelation, you actually have fewer true degrees of freedom. And so you're going to get these very spurious correlations. So what you're going to need to do is use some sort of spatial null. And there are basically in the field right now two broad families of spatial nulls, so-called permutation nulls, where you take a brain map, you project it to a surface, to kind of a sphere, you apply random angular rotations, you bring it back, and you have a new brain map that has the same values as before, but now the locations have been randomized while preserving spatial autocorrelation. The other type of parameterized null, or the kind of family of the other family of nulls are parameterized nulls, where you try to estimate spatial autocorrelation in the original data, and you then generate random data with approximately the same spatial autocorrelation. So this is the famous brain smash variogram matching method from Josh Burt. What we did, well, first of all, if you're interested in this sort of thing, we actually wrote a review on this recently on null models more generally in network neuroscience. But at the time that we started to get interested in this, we noticed that there had actually been at least 10 different methods in the literature floating around. So the original kind of spin null can be interpreted many different ways. These parameterized nulls can be implemented many different ways. And it wasn't clear to us how well they worked. So this is just an example of drawn kind of like a toy gradient going from front to back. And with these spatial permutation nulls, these are like different implementations of these spin tests. And there are also a few different kind of ways to do these parameterized data models. And they had all been used by different groups in the field. And it wasn't really clear to us which one is best, which one should you be using for different research questions and so on. And we wanted to know specifically how well do they control family-wise error. So I'm going to take a little kind of detour to show you exactly why these are so important. The way that we kind of came up to do this is as follows. So we take a brain map that we call X, another map that we call Y. You correlate them, you get a correlation coefficient. And what you can do is then take one of the brain maps and apply one of these nulls to them. So say that you're doing the spinning method. You take the Y map, you apply the spins. You can see what happens, these kind of areas here, what happens when you rotate into the medial wall. And each time now you can take one of these rotated null maps and correlate it with the original map, and you get a distribution of correlation coefficients under the null hypothesis that there's no relationship between the two maps. So what you can do is count the number of these null correlations that exceed in magnitude the original correlation coefficient, and you know, because we did this 1,000 times, you normalize by 1,000, you get p-value, and the number of times that you get p-values less than 0.05, that's your family Wise error. That kind of makes sense. Okay, so we were interested in two things then. How important is spatial autocorrelation, and is there any effect of the parcellation and the resolution of the parcellation when we do this? So the way that we kind of came up with a ground truth scenario is we placed Gaussian random fields on a grid, and then we projected them to the FS average surface. And that's what you can see in those brain maps over there. What I can do then is tune, there's this parameter I'm going to call alpha. I can tune the extent of spatial autocorrelation. I can get maps that look kind of like real brain maps. And the cool thing is now I can pick pairs of brain maps that have a desired correlation between them and also desired spatial autocorrelation. So this is just a sanity check. This is a histogram, which you probably can't see too well, but a histogram of the correlation coefficients between pairs of brain maps as I tune up the spatial autocorrelation. So you can see that when spatial autocorrelation is very low, you get a peak right at 0. So none of the maps are correlated with each other, but as they start to increase spatial autocorrelation, I start to get very wide tails, which means that I'm starting to get these spurious correlations that I'm talking about. So it works as I want. And now you're going to see what happens. So here, what you're seeing is the false positive rate on the y-axis. And on the x-axis, you're seeing the amount of spatial autocorrelation, and the curves are the different families of methods. This is for the FS average surface. So a few things to notice. Almost immediately, the spatially naive method, so the situation where you take two brain maps, you correlate them, you get the student's t value, you look up in the table, you know what the p-value should be. False positives shoot up into space almost immediately. So you get almost 100% false positives very quickly. With the other methods that are supposed to control spatial autocorrelation, they do so relatively well for low and moderate spatial autocorrelation, but high levels of spatial autocorrelation, they also suffer. This is true for the FS average surface. This is also true for the so-called Kamun parcellation, which is a subdivision of the Desikan-Kiliani, and for the Schaefer parcellation, which is a functional parcellation. And you see the results are very similar. What we also see more often than not is that the spatial permutation tests, so the so-called spin test and its various implementations, tend to be a little bit more conservative. So they tend to perform better compared to the parameterized nulls. So a few things that we can kind of conclude are, first of all, these naive nulls are completely inappropriate for significance testing. You should never be using them. Which spatial null you pick will depend a little bit on kind of your situation. So the spin tests are the most accurate, but they can only be used when you have surfaces. If you're working with volumes or you're working in subcortex or something like this, your only choice is to go with the parameterized nulls. But you're going to have greater type 1 error rates there. But the good news is, the silver lining is that these spatial nulls seem to perform identically across atlases, across resolutions, and so on. So it's not like you need to worry about matching, the null to the parcellation that you're using. Okay, so that was my very, very long digression into nulls, but I really like this topic and I think it's very important. The important thing to take away from this is that we've gone ahead and we've implemented all of these nulls, and they're in neuromaps, so you can use them. So that's kind of the tool. Basically, it's as easy as pip install neuromaps, and you can kind of get going relatively easily. Okay, so we did all that. Why do we do it? Well, we wanted to actually do neuroscience. So this is how we implemented some of these things. So I'll give you one example. You know, we have the map of all the maps of different neurotransmitter receptors. Obviously, the obvious question is, how do they map onto the kinds of cortical phenotypes of different diseases? So what we did was we queried the different Enigma consortia, and we got maps of cortical thinning for 13 different diseases. And so what you're looking at here are maps of, you know, T1 estimated cortical thickness, case control comparisons for these 13 diseases. Alzheimer's is not on there, so it hasn't been done yet. What we asked was the following. Could I take my 19 receptors and the PET maps for the distribution of different neurotransmitter receptors and use them to predict the map, you know, each of these maps? So the first thing that you see in the horizontal bar graphs are how well I can predict each of these maps of cortical thinning in different diseases. And what you're seeing is that we actually can predict most of them very well. These are very large R squares. We do so above and beyond the effects of spatial autocorrelation. As I discussed, the two diseases where we don't do so well are idiopathic generalized epilepsy. Not sure you would even expected a neurotransmitter receptor signature to be important there, and schizotypy, which is not really a disease, it's more of a trait. What you're seeing here so this is like a series of multiple regressions, basically, and now you're seeing the kind of the contribution to the overall model for the different neurotransmitter receptors. You probably can't see very well the labels, but I can kind of tell you. Basically, it's kind of like a nice lookup table now. You can say, you know, for a disease, what are the neurotransmitter systems that are affected? You know, this can guide my future experiments and so on. And you get a kind of a mix of things that we know very well, kind of textbook knowledge, and new things. So for instance, what you're seeing here is you see this stripe here. So this is the serotonin transporter. You see it being a hit for basically all the psychiatric diseases like OCD, schizophrenia, bipolar, and so on. That's what we would expect. And then sometimes you see hits that you don't necessarily think of when you see that disease, but then if you look in the literature, you do see support for it. One example is Parkinson's disease and the histamine 3 receptor, for which there actually is quite a large literature, especially in the animal models. So like I said, you know, I can kind of help you to guide new experiments, follow-ups, constrained models, and so on. I'll show you, maybe I'll just kind of skip through this part. It's a little bit long and maybe not super interesting for what we want to do here, because I think the next part will be better. This is another one where we're all, you know, our lab is more broadly interested in the link between structure and function, and so a lot of times function for us means dynamics. And so what we were looking at was, this is a paper in which we were interested in the link between fMRI connectivity and MEG connectivity. And so this is a map that shows you how the level of correspondence between MEG and fMRI in different brain areas. And the notable thing about the map is that you have high correspondence in primary visual cortex and primary sensory cortex, somatomotor cortex, and low correspondence in, you know, what you would call transmodal cortex. So it kind of follows this unimodal transmodal gradient. And one thing that we thought about was, well, maybe this has a little bit to do with how sensitive different areas are to neurovascular coupling, or how regionally heterogeneous neurovascular coupling is for different areas. So one thing that we could do is go back into neuromaps, and this time comparing things to the big brain atlas. You can see that the level of correspondence between MEG and fMRI tends to follow, well, has a relationship, a very specific relationship with cortical depth, and it seems to peak in areas that are most enriched and kind of that have the largest layer 4, at least in the big brain. And then we had this one very simple hypothesis, which was that you know, if it really has to do with neurovascular coupling, maybe this map should be related to some measure vasoconstriction and vasodilation. So we went into the Allen Atlas gene expression data, and we pulled a map for neuropeptide Y1R. This is the receptor for neuropeptide Y, and it's a vasoconstrictive. It has a vasoconstrictive effect. And what you can see is that there's this really nice anti-correlation between the two, meaning that at least part of these regional heterogeneities in the coupling between MEG and BOLT has to do with regional differences in vasoconstriction and neurovascular coupling. So those are just some examples, but the whole reason we kind of went into this field at all is this. So as Habib mentioned, I spent a lot of my time looking at networks and connectomes and so on. And when you do so, you start to believe that the brain looks like this. Representing the brain as a graph is a dimensional reduction step. It's a very good step because it allows you to kind of abstract away biological information about individual brain regions and helps you to focus on the connection patterns between them. But that's not what the brain really looks like. All areas are different. They're different in terms of their cytoarchitecture, gene expression, neurochemical signatures, and so on. And our modern kind of tools for looking at brain graphs don't really account for that. What you can do is you can take the kinds of maps that I just talked about, these various maps of these various microscale features, and you can actually superimpose them on your graph. So you can take a map of whatever microarchitectural annotation you have. You can look for for every node exactly where it lies on this map, and you can assign a scalar value or a vector of values to each and every node in the graph. And in network science, these are called colored graphs or annotated graphs. And in our case, because we're talking about connectomes, these are really biologically annotated connectomes, where now we have both a network structure, but also where we have information on our metadata on every node as well. And it's something that, again, we sort of reviewed recently, if anyone's interested. But the point is that we can take all of the maps that we just talked about, we can superimpose them on our connectomes, and we can ask questions about how the local biological features of a brain area are related to its network embedding. That's kind of the bigger question that we were seeking to address. I just want to give you a sense of how interesting things can get when you have biological annotations. Basically, everything that we've asked up until now of structural and functional networks, you can ask even more biologically meaningful questions if you have biological annotations. So, questions about homophily. Are two areas that have similar biological annotations more likely to be connected with each other than expected? For instance, you can plot, like, for these are here are points for areas that are physically connected to one another, and then what we're plotting is their scores on this particular annotation that's drawn here. And the idea would be that are areas that have a similar laminar differentiation more likely to be connected with one another. If you think about hubs, the field has been fascinated by highly or disproportionately connected nodes, where they are, and so on. You could also ask, if a node has many connections, does it also have different metabolic demands? Does it have a very specialized neurotransmitter signature? Does it have specialized electrophysiological dynamics, and so on? So you can relate various measures of centrality to various biological annotations. And then you can go on and ask a wide variety of other questions for, whether different modules or communities and networks have different biological signatures, and so on. So I'll kind of show you a few examples of this, but I still have time. This is like I said, our kind of primary interest in structure-function relationships. So one thing that we did a few years ago is we showed that if you were just to compare structural and functional connectivity across the brain, you get a map like this. This is kind of how well I can predict functional connectivity from structural connectivity. Again, it follows this unimodal transmodal gradient, where you have really good correspondence between structure and function in unimodal cortex and poor correspondence in transmodal cortex. So this is just going structure to function, or structural connectivity to functional connectivity. What we said was, well, look, the arrangement of neurotransmitter receptors should also modulate the link between, or the emergence of, functional dynamics on anatomical networks. So we have a second model here in which we're predicting functional connectivity from both structure and receptor information. That's what the map looks like. And now I'm showing you how well I can predict, again, function from structure for different brain areas. And what you can see is, so the line there is the identity line. And then what you're seeing is basically for the model where I've included receptor information, I now predict function much better, basically across all areas of the brain. And I do so significantly above the effect of spatial autocorrelation for most of them. So the idea is that by bringing in information about the underlying biology, I can actually make more veridical models. And that's something that I'll show you again now. This is not even that new anymore, but it's work that we were doing with Alain Dagger, who was interested. So this is very much, I think, in line with some of the work that you guys are doing. We were interested in how misfolded proteins spread on brain networks in different neurodegenerative diseases. And as you know, this is kind of like a dominant hypothesis in the field. What we were specifically interested was Parkinson's disease. And there, the misfolded protein is something called alpha-synuclein. So we were making these connectome-based, connectome-driven agent-based models where we would model individual molecules of synuclein, which would occasionally misfold, and when they came into contact with normal proteins, they could probabilistically cause them to misfold and aggregate, and they would spread from cell to cell. And when enough of them accumulate in a cell, either presynaptically or postsynaptically, they can cause cell death and atrophy. What we were doing is taking real, white matter, diffusion MRI derived connectomes. We were simulating the spread of these misfolded proteins, predicting patterns of atrophy, and then comparing this to actual patterns of atrophy in Parkinson's disease. That's kind of the idea. Why am I telling you this? Well, turns out that you can actually use biological metadata to make better models. What we did in our initial simulations was to say that, synuclein, we know, is continuously being synthesized and cleared in different parts of the brain. But what we were doing, at least initially, was to run models. So that I'm showing you here on the y-axis model fits, and in blue, here are model. Don't pay attention to the x-axis, just slightly different ways of reconstructing the network. But in blue, what you're seeing is how well the model does when I have equal, completely uniform synthesis and clearance for all brain areas. So you do relatively well. But in red, what we did was to say, well, actually, we know the gene that codes for synuclein. It's called SNCA. And we know the gene that codes for an enzyme to clear synuclein called GBA. So we went into the Allen Atlas and we simply said, okay, areas that have more expression of SNCA, they're going to synthesize synuclein faster. And areas that have greater expression of GBA, they're going to clear synuclein faster. Kind of very simple hypothesis. It doesn't actually involve an additional parameter or anything like this. But that's what you can see in red. Those models tend to perform a lot better. And in gray are null models where we control spatial autocorrelation and a few other factors. But again, the idea is by incorporating biological metadata into computational models, you can make more veridical models. You can also, just to kind of, I don't know if you're interested, but you can play this game for not just Parkinson's disease. We recently did this in FTD as well. You can pick different genes, different combinations of genes, and now you have also different subtypes of the disease, so you can do more or less the same thing. And then just kind of as a last thing that I wanted to show you is we said, okay, well, there are some people who say that the spread of disease in the brain is really going to be conditioned by the network architecture, so connectivity is really important. And then there's also always this other kind of competing, not competing, but complementary maybe hypothesis that areas are going to be selectively vulnerable to different types of pathology. So we said, okay, let's put them all side by side, head to head, and see which one actually does better. So what you're seeing here are different kind of predictors that we've constructed of connectivity. So just measures, sorry, this connectivity is here. So different measures of network embedding, connection diversity, and so on. And we're going to compare how well these predictors can predict disease patterns against these predictors here, which are predictors of local biological features. So everything from gene expression, receptors, excitation, inhibition, metabolism, synapse density, and so on. And so this is what you're seeing here. So again, I'm taking the 13 diseases from Enigma. I'm looking at how well I can predict each one of them using either the molecular, kind of local molecular predictors or these global connectomic predictors. And the main thing to notice here is that actually for most of them, I do a lot better when I use the local molecular predictors compared to the connectomic predictors. So, paying attention to the kind of local biology is really important. It turns out you can kind of see the paper. We actually show that they actually account for slightly different portions of variance. So there is a case to be made that both types of predictors, both connectivity and local biological information, could be used in tandem. And then I'm just going to skip this, but show you the very last thing, which is that now that we have these biological annotations, we can cross-reference them to structural and functional connectivity that we get from MRI, but we can actually also use them to build new types of networks. So this is the way that we one way that we can build a network of kind of an annotation similarity network that we're very familiar with. We take two brain areas, we look at their time courses of their neural activity, we correlate them, and we get a functional connectivity matrix. So we're correlating annotations across time. You can also do kind of the more old-fashioned thing, which is to take pairs of areas across participants and correlate their biological annotations. This is how we construct anatomical covariance networks, for instance, or how people used to do PET metabolic connectivity networks in the late 80s. So you can kind of get a participant similarity network. But what you can also do is if you have two brain areas and you have a vector of annotations, you can also correlate those as well, and you can get a network of how similar, again, are pairs of areas according to multiple biological features. And so what we recently did was to actually use all these data sets that we had to construct different types of biological interregional similarity networks. So correlated gene expression, how similar are your receptor profiles, how similar are your laminar distribution profiles, how similar is your dynamic PET metabolic activity. This is just good old functional connectivity. This is MEG-based, kind of phase synchrony, and then there's another one here that's derived from BOLD as well. But basically, the idea here is that when you construct these networks, you actually get brain networks or kind of interregional kind of connectomes that all have slightly different architectures. They are all picking up different interregional modes of kind of cooperation and communication. They all have slightly different patterns of hubness. They have slightly different modules, gradients, and so on. Whoops. And you can actually use them then to inform new models of disease spread, to kind of fit them to pit them against structural connectivity, functional connectivity that you get from MRI and so on. But anyway, so the idea is that you can really take these annotations and even make networks of them. So that's kind of all I had. I don't have a concluding slide because the point that I was trying to get across is really simple, and that is that by paying attention to the underlying biology, which we can because the data is out there, you can really make more accurate, more veridical, and potentially more meaningful models of the brain. And all the data is there for you, all the tools are there, and I think it's very simple to kind of get on board with some of this work. So yeah, that's all I had, and happy to take questions.
Thank you very much. So if you have some questions, go ahead. Okay. So this might be only for cortical regions, or you consider other.
So in our analyses, we didn't consider extracortical regions, but the maps that we provide are full volumetric maps. They include subcortex, cerebellum, everything.
We know that in many neurodegenerative diseases, you have a kind of directionally biased spreading of disordered species. Did you consider directionality in your analysis or not? Simple graph.
No, so like you know, in the PD work and in other projects where we've done this sort of thing, I mean, it's diffusion, MRI, reconstructed connectomes. We don't have any directionality. I suppose you could do and there are ways to try to put directions on, or to, you know, infer directions, but we haven't done that, no. I will say this, actually, I didn't show this, but again, together with Alain and some other people, we have done this synuclein spreading modeling in the mouse, because Abogen actually will get you the gene expression for the mouse. The LN Atlas provides you, you know, track tracing for the mouse. So we've actually shown that these agent-based models can really nicely predict the spread of misfolded. So these are you know, experiments where they purify synuclein fibrils and inject them in different locations, and then based on the anatomical connectivity plus local patterns of gene expression, you can make fairly accurate models of the. And again, now it's not, you know, it's not, you know, MRI-based cortical thinning, but really like immunohistochemistry, like based kind of readouts of synuclein deposition, and you can predict them very well. Yeah. So this is Shadi. I don't know if you guys know Shadi Rahal. He was, yeah.
And for predicting the spread of synuclein, we use a kind of differential equation, or we to say that the dynamical system.
Yeah, so we've done it both ways. Our initial implementation was an agent-based model, which is a discrete event simulation. So you're literally modeling every single molecule, every particle, one by one as they move through the network. We've done a differential equation-based version as well of this. It's an SIR model as well. Both are actually, I don't know if I had a link to it, but we do have like a not a toolbox, but like the code for that. But yeah, it works both ways. With a nicer thing with the agent-based model is we can implement more rules and more hypotheses, you know. So we've tended to stick with that. But when it's a simple kind of, you know, I want to know how a quantity spreads just based on the anatomy, then obviously like
Any other question? Okay, I have like a few, but let me just summarize. My first question concerns the spatial correlation that you're doing.
yeah.
Okay, so spatial correlation, what you're looking at is usually you use the correlation between two maps. And you do this kind of influence hypothesis H0 about the correlation. And we know that we can do any kind of permutation between the boxes, and we can got the null hypothesis that you talked about. My question, because this is something that has been addressed in different other areas, even in FMRI, I think Pierre Belair, his thesis was how we can correct from the spatial correlation to that the right map. This is one of his team. What about the work that people like Noël Crécy, who is in statistics, did in spatial correlation? deriving the law of the correlation itself. I mean, we can look at the spatial correlation and we can model the spatial correlation. And we can use this like Gaussian distribution.
Yeah.
So we call it Kriging, spatial Kriging, things like that, methods that we develop. So because I see something very similar of what you are doing.
Yeah, I can maybe try to contextualize that a little bit. I apologize for I went a little bit wild with the slides, but so maybe we can kind of go here. Yeah, so I'll say that the let's do this. Yeah. So actually, well, this method is the one that's become, or this family of methods has become really popular because it's so easy to do, and because it was, at least partly, I think, maybe a cultural thing. It was just invented more recently by a fairly powerful group at Penn and NIH. So people tend to use this, so it can maybe feel like people are inventing the wheel, or at least they're just kind of going with the newest thing. It's kind of the easiest thing to apply also. I think what you're talking about are kind of more broadly these families of, so it's kind of like non-parametric, whereas this would be parametric.
Yep.
I don't know the specific method that you're talking about, but these methods all kind of do the same thing, which is they try to estimate parametrically the spatial correlation in some way. So like, for instance, what's actually being shown here is a method that tries to estimate the variogram in the data in the brain map, and then it generates a just noise that's being then fitted iteratively to match that variogram. So that's exactly how this method works, and I'm sure that there are others much before it that came that were kind of the same thing.
It's exactly what I mean. Looking at the variogram, you can also look at the variogram in space and in time. And usually, what we did in fMRI and early technique is that we extract images, maps, and then we put a spatial model on the activity or whatever. But we can use the variogram in space and time at the initial data set. And because in this case, there are a few models who did not separate between space and time, which all the techniques that we use do, separate between space and time. And we can have this variogram who merge between space and time. It's a non-linear approach. And this non-separable variogram is also a variogram who has a good properties that we can generate, things like that. So I don't know if it's this increase, I mean, the sensitivity of the method or whatever, but it seems to me like very close to.
Yeah, that's really, really interesting. I mean, like, as I showed you, these methods are still not perfect. They're far from perfect. So there's like a lot of room for improvement. Actually, they're kind of new papers all the time trying to do this. The nice thing is, I mean, I didn't invent either method, but I think the nice thing that we did was we came up with a good way to actually quantify false positives. So I think like new methods, if they are implemented, like basically we kind of have the entire framework set up. So it would be cool to know if there's maybe something from related literatures that works.
I have another question about the structures and the function. So this is like one of your main topics, and you did several works before. I remember that you did some work looking at simply comparing between structural connectivity and functional connectivity, showing that, okay, there are a good mapping between the two in several networks, and
Mhm.
model networks seems like there is large differences between the two. Do you think that if following your research program and following what you are doing with the neurotransmitters now, because the neurotransmitters are going probably to play a role, this is what we did, and we show that with neurotransmitters you see a more correlation, can explain more. Can we come back to this first result of and adding the neurotransmitter to see if some network correlate more if we add this part of information?
Yeah, that's exactly what we see. Yeah.
What
Yeah, that was that. Yeah, so I mean, yeah, so that was actually the really the reason I started to become interested in local biological features at all, because we're often seeing these regional heterogeneities in structure function coupling. And we thought, well, they must have to do with something about the circuit properties of the nodes themselves in the neural population. So we started to do this. You see an increase across the board when you include neurotransmitter receptors, both in unimodal cortex and in transmodal cortex. Obviously, in transmodal cortex, you already have such poor prediction of function from structure that it's easier to improve. There's more room for improvement. But you see the enhancement all across the board. We actually, in that paper, Justine's neurotransmitters paper where we did this, she actually has a map that shows exactly which I did not show just for simplicity, but it's a map of exactly which areas tend to benefit most from including receptor information when you're trying to predict function from structure. And the interesting thing there is honestly, we don't really know whether that map is reflecting something about those areas just having more maybe neuromodulatory input from the brainstem, and maybe that's why they're more strongly subject to these neuromodulatory influences, or if those if the information about neurotransmitter receptors is somehow filling in gaps or missing information from diffusion MRI, which could also be the case, because a lot of the places where we see them, again, you're gonna have to take my word for it. A lot of the places where we see improvement are in medial temporal areas and things like that. So it could be that we're just doing less well there with diffusion anyway, and that's why we were seeing lower structure function coupling. And now when you add in the receptors, maybe you're kind of plugging in the gaps a little bit. But which is actually, I'll just say the last thing is that's what we are now doing a lot of work on incorporating the brainstem into these connectome models. So we have on preprint where we found an atlas of about 70 different brainstem nuclei, and we have their functional connectivity to the rest of the brain. So now we have these connectomes that are not just cortical but really subcortical and very specific about the particular nuclei populations that are represented. So, yeah.
And you did this correlation between MEG and ball signal. So which part of MEG are you correlating to ball signal?
Oh yeah, so yeah, I breezed over that. So that's yeah. Oh, sorry. Yeah, so this is a map where you take, for one brain area, you take its functional connectivity to the rest of the brain. So you kind of have a vector. And then for that same brain area, you take its, we did this many different ways, because we know that different people like to do things differently, and so the reviewers wanted it. We did amplitude envelope correlations for the different kind of canonical electrophysiological bands, and we also did space synchrony as well. So we did it kind of both ways. We did it per band. There are also interesting differences among the bands. It's kind of more of a summary map, but by and large they tend to perform around the same. So yeah, But the actual analysis is done per band.
Any other question?
We don't really have time. There's no way to
The-
go to.
All the group, okay?
Well,
Okay, so let's have the last question. Yeah, go ahead. No, last question. No? No question. Okay.
we.
Thank you.
Thank you.
Thank you.